Okay, so today we're going to look at A2 homeostasis and the kidney. Okay, in terms of homeostasis, that is about keeping things balanced. So you can see this seesaw is often used. So we want a nice, even, balanced seesaw, everything controlled. Okay, so homeostasis is about controlling things within the body. So we have to control things, as you can see here, like the amount of water, the concentration of water within the blood, the body temperature, things like the amount of glucose, etc., within the blood as well. Okay, it requires receptors to be able to detect a change. If that change occurs, then a control center will lead to some sort of effector and responding to that change to bring about a corrective mechanism. Okay, so we want to, if things get unbalanced, then we need receptors to detect that. We need a control center to make a decision, most often the brain, to make a decision to release, usually a hormone, which is then released by the effector, and that will bring things back down to normal again. Okay, it also requires negative feedback, which means that if you think about these corrective actions, that's switching a tap on. The negative feedback is then the normal level has been brought back. So we need to switch that tap off. Okay, so it reduces the corrective action or the reduction of, for example, a hormone being produced, which means that things have been brought back to normal again. Okay, so the kidney is one organ which deals with homeostasis of water and also glucose levels. Okay, and in terms of the gross structure of the kidney, we have two kidneys, left and right, obviously. The aorta, which brings blood from the heart, brings blood into the kidney through the renal artery, which you can see here. The kidney acts as I suppose as a bit of a filter, so it takes out the useful things out of that blood and reabsorbs them. Any waste materials like urea, etc., excess water, excess amino acids, that type of thing, will be got rid of within the kidney. That passes out then through the renal valve and brings blood back to the heart. So that will bring the useful materials back. Any waste material will pass down the ureter and be stored in the bladder, ready for excretion. Okay. If we take a look at the sort of detailed structure of one kidney, the outside of the kidney, that sort of protective layer on the outside is known as the cortex. Inside of that, you have the medulla, sort of in the middle, and then you have the pyramids here of the medulla, or pyramids of the pelvis, which then leads down into the ureter. Okay, and the ureter, as we said, brings things down into the bladder. Okay, in terms of the division between the cortex and the medulla, you can see here on screen. So the cortex, that outer region, contains the glomerulus and Bowman's capsule. It contains the proximal convoluted tubule. The descending limb of the loop of Henle passes down through the cortex into the medulla region. The ascending limb, that turns then at the apex there, the ascending limb passes back up through the medulla into the cortex. You have your distal convoluted tubule, and then the collecting duct, which brings the waste material down to into the pelvis, passes back down through the cortex, down through the medulla, and then will pass into this pelvis region and down into the ureter. Okay, you'll also see the very good blood supply around different parts of the nephron, or all parts of the nephron here. That's to do with reabsorption, as we'll look at in a second. Okay, so you can see here, first part of this, there's your gross structure, so cortex around the outside, medulla in the middle. There's the pyramids that you can see as well of the medulla. So there's your glomerulus and Bowman's capsule, proximal convoluted tubule, loop of Henle down into the medulla, distal convoluted tubule, collecting duct leads down into the medulla and then down into the pelvis and out through the ureter. Okay, if you can organize yourself to know the structure of the nephron and what happens at each stage, that's half the battle. Okay, so the first part, the glomerulus and Bowman's capsule, is to do with ultrafiltration. Selective reabsorption, as that suggests, certain things are taken out of the filtrate, or the liquid passing through the nephron and back into the blood. happens in the proximal convoluted tubule, and then osmoregulation, or controlling water content within the blood, is really involving the rest, which is the loop of Henle, a little bit of the distal convoluted tubule, and the collecting duct. Okay, so the first part, ultrafiltration, as we said, occurs in the glomerulus and Bowman's capsule. Okay, so blood leads through into the glomerulus, through the afferent arteriole, so this will have come from the renal artery. It passes in through here. This is very winding, this glomerulus, very long, very highly folded, and that increases the surface area and then passes out through the efferent arteriole. You'll also notice that the afferent arteriole is wider than the efferent arteriole. That leads to a pressure buildup inside the glomerulus. If you think of a wide river where the water is moving quite slowly, going down into a narrow river, the water needs to move faster, so it builds up the pressure. Okay, you can see the expanded diagram here of this section. So this is the blood capillary from the glomerulus. You can see it's only one cell thick. Okay, it's got squamous epithelium here, which is a flattened epithelium, which has got gaps in the center. Okay, then you have the basement membrane, which is sort of the first part, if you like, of the Bowman's capsule, and below that you've got podocyte cells, which are involved a little bit in ultrafiltration as well. Essentially, the effective filter here is the basement membrane. Okay, so it decides what passes from the glomerulus into the Bowman's capsule. Okay, and generally, everything that passes from or in the blood will pass through. Okay, apart from larger blood vessels, okay, like in red blood cells and white blood cells, and also any proteins, any plasma proteins that you find in here, they're too big to pass through that selective filter or that basement membrane, so they stay within the blood. Okay, anything that passes through into the Bowman's capsule from now on is called a filtrate. Okay, another little diagram here you can see just showing that afferent arteriole leading in, highly folded here, long path to pass down to that blood allows a lot of substances to pass into the Bowman's capsule. Okay, again here you can see that the capsule, you can see there's your blood vessel, there's your squamous epithelial cells, there is the basement membrane, these are the podocyte cells. We'll take a look at the structure at the moment. You can see here the different structures or the different substances, urea, water, and glucose, protein, sodiums. The only things which stays, or the only substance which stays within the blood are the larger proteins. Okay, everything else will pass down into the filtrate, and we'll see how that happens in a moment. Okay, there's an electron microscope diagram, so you can see the outsides there of the Bowman's capsule. You can see the blood glomerulus, so the capillaries leading inside of here as well, highly folded, as we say. There's the podocytes. Podocytes are a bit like a hair clasp, which surrounds that basement membrane, or surrounds the capillary, if you like, that you can see there. Again, it helps a little bit with filtration or ultrafiltration down through the basement membrane. Okay, in terms of what enters or how it enters, there is a concentration gradient here, which you can see. There's a concentration gradient of glucose, high concentrations in here, lower concentration in the Bowman's capsule. So about 50% will pass through into the Bowman's capsule. Okay, some may, when it builds up in here, there is a concentration gradient back into the glomerulus. Okay, but because of that pressure buildup, because the afferent arteriole being wider than the efferent arteriole, that buildup of pressure, the net movement of solutes is from the glomerulus and into the Bowman's capsule. Hence that ultrafiltration. So it's small things that are filtered through, big blood vessels that you can see here, larger proteins that would be in the blood capillary as well, don't pass through. Okay. The next stage then is looking at selective reabsorption, and this occurs in the proximal convoluted tubule, which you can see up here. So things have passed through, your glucose, urea, etc., have passed through into the Bowman's capsule. Now they're moving through the proximal convoluted tubule. Okay, and anything which is useful to the body, we would want to reabsorb that. Okay, so you can see here that things that pass through, here you go, there's urea, water, proteins, sodiums, etc. They will pass through into the Bowman's capsule, and then useful things we'll want to reabsorb back into the blood. So you can see proteins and glucose, any water will want to pass back into the blood as well. Okay, the cells of the proximal convoluted tubule you can see here. Okay, these are cuboidal epithelial cells. They're highly folded or have microvilli on their surface. That will increase the surface area, as it says there, to help with reabsorption. This is what the lumen, where the filtrate will pass down through. Okay, within the cells as well of this proximal convoluted tubule, we have large amounts of mitochondria. Okay, and that, as you can see there, provides ATP for active transport. At the bottom of these cells as well, beside where you would have your blood capillaries, you have infoldings, and again that increases the surface area, which makes it easier for diffusion to occur. Okay, there's also proteins within the membrane here, which help with facilitative diffusion and active transport as well. Okay, so generally you'll have these examples of glucose, amino acids, urea, and things passing through here. 50% of them will pass by diffusion, from high concentration to low concentration, into these cells. Okay, and essentially that's your glucose and your amino acids. We're looking at a little bit of water as well will pass through. In order to absorb all of the glucose and all of the amino acids, which is essentially what the body wants, because those are useful materials, you want those to go back into the blood, then active transport occurs. So these glucose and amino acids are actively transported out of the cells and into the capillaries, which means then more will follow by diffusion. And you can see that at the top here. If we take a look at that, you've got cells passing down through these highly folded layer at the top of the proximal convoluted tubules, and then you can also see here you've got facilitated diffusion and active transport of certain substances passing into the blood capillary and then taken away, which maintains that concentration gradient. Okay, there again you can see those cells, so that the filtrate will pass through, highly folded layers, so good for diffusion. Glucose, sodium, water, things like that will pass through these carrier proteins. You've got the active transport then of glucose and proteins into the blood to make sure that all of that gets reabsorbed in the proximal convoluted tubule. Okay, so ATP generally used down in these invaginations or foldings at the bottom, allowing all of the glucose and all of the amino acids to be transported back into the blood and reabsorbed. Okay, the next bit of this then is looking at the loop of Henle. As we said at the very start, the loop of Henle and distal convoluted tubule and collecting duct are really to do with osmoregulation. So that's to do with keeping the concentration of water within the blood the same. Okay, so the descending limb of the loop of Henle is permeable to water. The ascending limb of the loop of Henle is not permeable, or it's impermeable to water. Okay, but it does allow certain ions to pass through the walls. Okay, and these ions are actively transported out of the ascending limb into the medulla. Remember, this stretches down into the medulla region. Okay, so when these ions are actively transported out, your sodium chloride ions, that's essentially what makes up salt. It makes this area of the medulla very salty or very high in solutes. Okay, and what that means is as the filtrate passes down through the descending limb, water will pass out of the filtrate by osmosis. Okay, so it tries to dilute that highly salted medulla area. As the water passes out, this filtrate becomes more concentrated. Okay, so water, it's losing water, so therefore it's becoming more concentrated. So imagine if you had a cup of tea with a sugar in it at the top here, and then it lost some water, so you boiled some of the water off, it would become more sort of syrupy or more sugary. So it's more concentrated. And the most concentrated or hypertonic part of the filtrate is at the bottom of your loop of Henle. As it passes back up through the loop of Henle, then you've got some of these salts passing out, your sodium chloride ions. That makes our filtrate less concentrated as it moves up the ascending limb. Okay, so that's often referred to as the countercurrent multiplier. So these are moving in opposite directions, and it's getting more concentrated and less concentrated as it moves down the way and back up the way. The whole point of this is to make the medulla region highly concentrated in solutes, so therefore water will then pass out of your loop of Henle and be reabsorbed into the blood. Okay, it's also allowed to allow water to pass out of your collecting duct, which means that more water is reabsorbed as well, which is essentially what osmoregulation is about. Okay, and as water is reabsorbed from the loop of Henle, all of those waste materials that stay in here, urea, etc., other ions that you don't need, that becomes more concentrated, and that will pass down the ureter to the bladder, and that becomes hypertonic or highly concentrated urine. Okay, and you can see that in this little diagram as well. So you've got active transport of sodium chloride ions in the ascending limb of the loop of Henle. That decreases the water potential of the medulla region and makes it very salty. That will draw out water as it moves down through the filtrate, down the descending limb. That makes the filtrate more concentrated, and then you've got some of those ions will pass out as it moves back up the ascending limb again, which makes it less concentrated. So if that is a very highly concentrated medulla region, it means that more water will pass out through the collecting duct to be reabsorbed, which means you get more concentrated urine. Okay, and we can see that in, sorry, this little animation here, where for the collecting duct to become more permeable, it needs to open up little aquaporins, which are little pores within the walls of the collecting duct. And we do that by secreting a hormone called ADH, or anti-diuretic hormone. Diuretic is something which makes you lose water. So tea, coffee, alcohol, fizzy drinks, all examples of diuretics. They make you go to the toilet more, and your blood or your water concentration in your blood decreases. Okay, that's detected by receptors in the brain. Okay, and ADH is made in the hypothalamus and is secreted then within the brain. And as it's secreted, or when that makes its way down to the kidneys, that will cause the pores in the collecting ducts to open up. And because this tissue is very salty in the medulla, then water is drawn out of the collecting duct, so it's reabsorbed in the collecting duct. It'll pass into the blood. That causes the water concentration in the blood to increase. So that's the corrective mechanism that we talked about at the start. When that returns to normal in terms of the water concentration of the blood, then you get negative feedback occurring. This secretion of ADH will stop, which then reduces the permeability of the collecting duct walls to water, which means that less water then is reabsorbed again. Okay. When the water is reabsorbed, that means there's less water in the urine, which means that it'll be more concentrated. So if you do exercise, if you don't drink very much during the day, you'll be secreting ADH. So the urine that you do produce will be very yellow in color, and you'll have small quantities. Okay. Whereas if you do drink a lot of water over the day and you don't do much exercise, then your blood concentration or water concentration within the blood should be fine. You don't secrete as much ADH, you don't reabsorb as much water, So you produce larger quantities of less concentrated urine. So it's a more clear solution. Okay, and here you can see there's the normal. Think of that seesaw balanced. That's your normal level. If, let's say, you do some exercise, that will lower the concentration of water in your blood. Okay, so it decreases the solute potential or decreases the water potential. Then you'll produce more ADH. You'll reabsorb more water from the collecting ducts. You produce small volumes of highly concentrated urine, and then that will bring it back up to normal. Okay. If you drink loads of water during the day, that will increase the water potential or solute potential. You don't produce as much ADH. You reabsorb less water, and you need to go to the toilet more often. And the pee that you produce is less concentrated or more dilute, and that brings it back down to normal as well. And the final thing to look at is just there's an electron microscope image of some of the collecting ducts. Okay. Hope you find that useful. Thanks for listening.